So a genetic primer. So this is about PCR, polymerase chain reaction, or how we make copies of DNA that we need in the lab. So if you look at the cartoon on the left, you see the thermocycler, the machine. Ours is purple, but we have one that looks like this. And you see the first part of the cartoon is a blue, very short section of DNA. Right? You can see the double ladder. And then you can see that it says 98 degrees Celsius, which is really hot. That's like 200 degrees Fahrenheit. And you can see that the two sides of the DNA have fallen away from each other. So we call that denaturing, right? When you get it hot and it breaks apart. Then you see the next slide, you see that there are two little yellow sections of DNA, one on one end of the top strand and one on one end of the bottom strand. And those are the primer. It's a short sequence of DNA that brackets, and by bracket I mean like you do in algebra where you put the brackets on the outside of the equation. It brackets on the outside the part that you want to copy. And then you can see in the next part of the cartoon, there's that little donut-looking enzyme is adding little red bases to the left of the yellow one on the top and to the right of the yellow one on the bottom. And so that is, if you look below, making our copies. And so we're going to start with, in the first round, we're going to take one piece of DNA and make two. But if you keep going, right, by the 30th cycle, which takes about 3 hours, you have 2 billion copies of that segment of DNA. And we can actually do that in our classroom because we have the stuff. But again, a primer is a short, single-stranded piece of DNA that we use in the PCR reaction to kind of tell the machine what to copy, what part of the DNA we want to copy.